Hi biology students, today we're going to do the Floating Leaf Lab Part 2. Today we're going to be testing the effect of temperature on the rate of photosynthesis. There is a really helpful ed puzzle in today's folder, and you can watch that at any time, but I highly recommend you watch it before you write the CER at the end of today's document, because it will be really helpful in helping you to write a good CER. Remember from last class when we did Floating Leaf Lab Part 1, we can measure the rate of photosynthesis by using basically a hole punch and cutting out little leaf discs like you see in this beaker, removing the air from them, allowing them to sink to the bottom in water, and then recording the time it takes for them to float to the surface of the water. Because if photosynthesis is happening, they are producing oxygen bubbles which attach to them and help them rise to the top. Pretty cool. So last class we asked, how does the presence of carbon dioxide affect the rate of photosynthesis in spinach leaf discs? You were asked to write a CER based on the data you collected. I'm now going to show you a sample CER. In general, when you write a CER, your claim is a statement, usually just one sentence, that answers the research or lab question based on the data that you collected. Your evidence is a statement, usually a couple of sentences, that uses numerical data to support your claim and also to explain the trend or pattern that the data shows. And then your reasoning is where you bring in the science and you use the scientific principles involved to explain how your evidence supports your claim and why it all makes sense. And this was the graph that was generated by the data you collected for Floating Leaf Lab Part 1. It shows that in beaker A, where there was carbon dioxide gas, the rate of photosynthesis was faster. It took less time for a specific number of leaf discs to rise to the top of the water in the beaker, versus beaker B, where there was no carbon dioxide gas. It took more time for the same number of leaf discs to rise to the top of the water in the beaker. So based on that data, you can write a claim that would sound something like this. Carbon dioxide increases the rate of photosynthesis. We are always looking for you to use numerical data, which means numbers. So beaker A, which had 2 scoops of baking soda, had an average time of about 240 seconds, while beaker B, with no baking soda added, had an average time of about 305 seconds. And then here's my trend statement. The leaves in beaker A with carbon dioxide rise faster than those in beaker B with no carbon dioxide. My reasoning is going to explain how my evidence supports my claim. We know that photosynthesis requires carbon dioxide, so when we add it to beaker A, the leaf discs can do photosynthesis and rise up. The graph shows that beaker A leaf discs took less time to rise than beaker B leaf discs. We can therefore conclude that beaker A had a faster rate of photosynthesis as a result of the baking soda added, which provided a source of carbon dioxide gas. Notice I don't have any additional information, and I'm clear, concise, and not confusing. Today, after you collect data for Floating Leaf Disc Part 2, you will write a CER, and you can use these slides as an example of how to format your CER using the data you collect today. So today, you are going to consider how, when light is absorbed by photosynthesis, the energy is used to change the carbon dioxide into an organic molecule called glucose, which is a sugar. We've learned the formula for photosynthesis. Carbon dioxide plus water plus sunlight makes glucose and gives off oxygen as a waste product. This is what it looks like in the language of chemistry. Your background information says that we can measure the rate of photosynthesis by cutting out leaf discs, removing the air from them, allowing them to sink in water, and recording the time it takes for them to float to the surface of the water. I showed you this a few minutes ago. We add baking soda as a source of carbon dioxide, and the leaf produces oxygen as a waste product of photosynthesis. So today we're asking a slightly different question than we asked in part 1. We are asking, how does temperature affect the rate of photosynthesis in spinach leaves? We are going to test this at 2 different temperatures, 20 degrees Celsius and 40 degrees Celsius. We want to know, how does temperature affect the rate of photosynthesis in spinach? This table should look very familiar. You did the exact same table for part 1. I am now going to set the timer for 10 minutes so that you can write a hypothesis, identify the independent variable in today's experiment, identify the dependent variable in today's experiment, and list 2 to 4 constant variables that you need to be aware of, and make sure they are the same in beaker A and beaker B in order for your data to be valid.